Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be explaining fatty acid oxidation, and while I explain fatty acid oxidation, I will be going over mobilization of fatty acids from adipose tissue, transport of fatty acids from adipose tissues to the peripheral cells including liver, and then a transport of long chain fatty acids into the mitochondrial matrix via carnitine shuttle mechanism. And then I will be going over some of the disorders that are associated with carnitine shuttle mechanism, and that is carnitine deficiency, carnitine palmitoyl transferase 1 deficiency, and carnitine palmitoyl transferase 2 deficiency. Let's get into the details. Long chain fatty acids are the principal fuels for energy which will undergo oxidation to give ATPs, especially in the form of acetyl-CoA. Acetyl-CoA will go into TCA cycle and eventually making NADH, FADH, and that will go into electron transport chain to give energy. Now, when we are in fasting condition, especially like overnight fasting or between the meals or during exercise, in these conditions, energy needs of our body is partially met by oxidation of long chain fatty acids into acetyl-CoA, and then acetyl-CoA it feeds into TCA cycle. Now, the long chain fatty acids are principally they are stored in the adipose tissue as triacylglycerol. Now, the triacylglycerol is the one that will store majority of the fatty acids in our body in adipose tissue. Now, fatty acids from the diet and from the liver, they are carried all the way to the adipose tissue either in the form of chylomicrons or VLDL. During fed condition, dietary fatty acids are carried from the intestine to the adipose tissue in the form of chylomicron, because tags are predominant type of fatty acids present in chylomicron, which is 90% of chylomicron. Whereas under fed condition, where excess carbohydrate which is converted into fatty acid and esterified into triacylglycerol, and this triacylglycerol from the liver, it will be carried to the adipose tissue via VLDL, very low density lipid. Now, see the type of fatty acids that are found in the adipose tissue attached to the triacylglycerol. It depends on what is the dietary source of our lipids. So, animal fat, generally it contains a long chain fatty acid which is saturated, and also we will see some of the monounsaturated fatty acid. Whereas a plant source, that is vegetable oils, usually they contain polyunsaturated fatty acid. Now, depending on the type of food a person takes in, so if it is a predominantly animal based diet, so tags will be containing more of saturated and monounsaturated fatty acids. If it is predominantly a plant based diet, so tags in the adipose tissue may contain predominantly polyunsaturated fatty acid. Now coming to, that's about long chain fatty acid. Coming to the medium chain fatty acids, so the medium chain fatty acids are principally found in dairy fat, maternal milk, and also in some of the vegetable oils. Now when you consider all this, so the major fatty acids that are undergoing oxidation in our body, they are palmitic acid or palmitate, which is a 16 carbon fatty acid, and in fact palmitic acid is the most common fatty acid that is synthesized in our body. And then oleic acid, which is a monounsaturated fatty acid having 18 carbons with 1 double bond. Stearic acid, which is a saturated 18 carbon fatty acid. In fact, stearic acid is the most abundant fatty acid in the nature. And also linoleic acid or linolate, which is an 18 carbon 2 double bond fatty acid. So these are the fatty acids which are the major fuels for our body during overnight fast, between the meals, and also during exercise, and also when the fasting continued for a long period of time and eventually going into starvation. Now, the triacylglycerols, which are present in the adipose tissue, they need to undergo lipolysis to release fatty acids, so that fatty acids can be carried from the adipose tissue all the way to the peripheral tissues like heart, skeletal muscle, liver, and all other organs that need energy during fasting or between the meals or during exercise. So how that is done? So whenever we go into fasting condition or when the person is exercising, so it can be either glucagon binding to glucagon receptor, or epinephrine binding to epinephrine receptor. So here in the figure, epinephrine is written as adrenaline, that's the older terminology. So glucagon binding to glucagon receptor, epinephrine binding to epinephrine receptor. So these receptors are G protein coupled receptors, so they will be activated by separation of alpha subunit from beta gamma subunit. Now the alpha subunit bound G protein, retake. Now the alpha subunit with the GTP, which is separated from beta gamma subunit. So the alpha subunit bound GTP is going to activate an enzyme which is present over cell membrane called adenylate cyclase. Now the adenylate cyclase is going to convert ATP into cyclic AMP as it is shown here, and the cyclic AMP is going to activate enzyme called hormone-sensitive lipase in the adipose tissue. And this hormone-sensitive lipase, what it does, now increase in cyclic AMP, it is going to increase the activity of protein kinase A enzyme, and this protein kinase A enzyme, it is going to phosphorylate a hormone-sensitive lipase enzyme in the adipose tissue, and phosphorylated form of hormone-sensitive lipase, it's an active enzyme, and what it does, it is going to degrade triacylglycerol into a free fatty acid, converting it as monoacylglycerol, and this is from the like the first carbon fatty acid is released there. I will write it as fatty acid number 1 coming from the first carbon, and then a diacylglycerol is undergoing a reaction catalyzed by hormone sensitive lipase, releasing one more fatty acid present at the third carbon. So the first carbon fatty acid is released first, and the third carbon fatty acid is released, and now the remaining molecule is 2-monoacylglycerol, 2-mag. And now this 2-monoacylglycerol is now catalyzed by 2-monoacylglycerol lipase enzyme, and this monoacylglycerol lipase, it is associated with a lipoprotein lipase enzyme. Now the lipoprotein lipase enzyme, it is in association with monoacylglycerol lipase enzyme in the adipose tissue capillaries. So basically monoacylglycerol lipase here, it is going to degrade a fatty acid present in the second carbon of monoacylglycerol and release another free fatty acid coming from the second carbon, and a glycerol molecule is released. Like this, a triacylglycerol, as you see, TAG, it has 3 carbons and 3 fatty acids attached, and these 3 fatty acids are released upon lipolysis. Now these are all free fatty acids, and these free fatty acids need to be carried from the adipose tissue in the blood all the way to the peripheral tissue. Now depending on the type of these free fatty acids, so the free fatty acids that are released here are usually long chain fatty acids, and the long chain fatty acids, they contain carbon atom more than 12 and less than 22. So that means long chain fatty acids are water insoluble, means hydrophobic, so they need to be carried by binding to certain molecules that can keep them soluble while they are transported in the blood. Now let's look into that. Now what I have explained in the previous slide, I have written here just as a recap. So let me quickly recap the previous figure explanation that I did. So glucagon or epinephrine binding to their corresponding G protein receptors, it is going to activate adenylate cyclase, thereby increasing CTKMP, and that activates protein kinase A enzyme, and that protein kinase A enzyme is going to activate hormone-sensitive lipase enzyme, which is going to break triacylglycerol into fatty acid, two fatty acids, and a monoacylglycerol. And monoacylglycerol is lipase enzyme is going to break a monoacylglycerol into another fatty acid and a glycerol molecule. Now, here in this figure, it shows an overview of fatty acid oxidation, starting from how all these long chain fatty acids which are coming from the adipose tissue. Now, in the previous slide, as I said, lipolysis that is going on in the adipose tissue, releasing usually the long chain fatty acids that are bound to a triacylglycerol. And these long chain fatty acids, as I said, these are water insoluble hydrophobic molecules. So they need to be carried from the adipose tissue in the blood to all the peripheral tissues. So since long chain fatty acids are water insoluble, they will be attached predominantly with the albumin. Albumin is a most abundant plasma protein, and albumin acts as a carrier of long chain fatty acids from the adipose tissue to the peripheral cells. And that is what is shown here in the figure. As you see, albumin carrying long chain fatty acids. Now the long chain fatty acids over the outer like a plasma membrane, so albumin is separated and the fatty acids can undergo either simple diffusion or it can use a saturable system to enter in. Once it enters into the cytoplasm, in the cytoplasm, these long chain fatty acids are momentarily binding with a fatty acid binding protein, and later these fatty acids will be activated over outer mitochondrial membrane and they will get into the intermembrane space. And in the intermembrane space, long chain fatty acids are esterified with the carnitine to become fatty acyl carnitine, and then fatty acyl carnitine is transported into the matrix of mitochondria, and inside the matrix of mitochondria, fatty acyl carnitine is reformed back into fatty acyl-CoA and carnitine is separated, and carnitine go back into the intermembrane space again. And that particular aspect there, we are going to study that as carnitine shuttle mechanism. Now, once your fatty acyl-CoA is there inside the mitochondrial matrix, it will undergo beta oxidation, meaning a beta carbon in a fatty acid will undergo oxidation. So, during this process of beta oxidation, there will be release of FADH2, NADH plus and an acetyl-CoA molecule, and in each beta oxidation spiral, this type of molecules will be released, and depending on the number of carbons in a fatty acid, there will be multiple beta oxidation spiral going on to completely oxidize a fatty acid into acetyl-CoAs and while releasing NADH and FADH2 molecules. Now, once we get acetyl-CoA after fatty acid oxidation, acetyl-CoA has got two fate in the matrix of mitochondria. One is it can go into TCA cycle to give 10 ATPs per molecule of acetyl-CoA, or especially in the liver, acetyl-CoA can be diverted into ketone bodies, and these ketone bodies will be secreted out of the liver into the bloodstream, and the ketone bodies can be used by other tissues as the energy sources. Now, this is the overview of fatty acid oxidation. So, this is what I have written here briefly with the previous figure, whatever I have explained, just as a recap one more time. So, fatty acids are carried to the peripheral tissues by binding to albumin, and then these fatty acids which are entering into the cytoplasm through plasma membrane, and from there they need to go into the matrix of mitochondria. Now, it depends on the carbon length of fatty acids. If the fatty acids are long carbon fatty acids, long chain fatty acids, usually long chain fatty acids, they contain carbons more than 12 and less than 22, meaning 13 to 21. So, they use carnitine shuttle mechanism to get into the matrix of mitochondria, and they are activated over the outer mitochondrial membrane. Now, if the fatty acids that are reaching to cytoplasm are medium chain fatty acids or short chain fatty acids. Medium chain fatty acids have carbon between 6 and 12 and short chain fatty acids have carbon less than 6. These fatty acids, they directly enter into the matrix of mitochondria through inner mitochondrial membrane and they are activated in the matrix. Now the activated fatty acids, whether they are long chain, medium chain or short chain fatty acids, they will undergo beta oxidation. Note that beta oxidation is for even number carbon fatty acids and also saturated one. Even number carbon saturated fatty acids undergo beta oxidation, whereas if the fatty acid has got unsaturation, so initially that unsaturation has to be converted back into saturated form before they undergo beta oxidation. Anyway, so once fatty acids undergo beta oxidation, what we get is acetyl-CoAs and NADH and FADH molecules. Acetyl-CoAs, they go into TCA cycle or in the liver, they can go towards ketone body formation. NADH and FADH will go into electron transport chain to give ATPs. And this is what is the overview of fatty acid oxidation. So we are going step by step in understanding how fatty acids are mobilized from the adipose tissue. So the mobilization of fatty acid from the adipose tissue, we have a role of glucagon and epinephrine, adenylate cyclase, cyclic AMP, protein kinase A, and then hormone-sensitive lipase, and then we have monoacyl glycerol lipase and releasing fatty acids. If the long chain fatty acids not soluble in water, so binding with albumin to be carried to the peripheral tissues. Short and medium chain fatty acids, although they are not a predominant type coming from adipose tissue, but just in case if there are medium chain fatty acids especially. So short and medium chain fatty acids, they are water soluble, so they just come in the blood being soluble, right? So when they pass through the plasma membrane, so they are activated over outer mitochondrial membrane, especially the long chain fatty acid. Short and medium fatty acids, they just go through the mitochondrial membrane into the matrix, so they are activated there. So whether it is short, medium or long chain fatty acids, all of them undergo beta oxidation to give acetyl-CoA, NADH and FADH. So all this information we are going a stepwise manner. So let's go into transport and cellular uptake of free fatty acids, which I have already explained to you earlier. So MCFAs and SCFAs, water soluble, so they are freely transported. LCFA, they are hydrophobic, they have carbons more than 12 and less than 22. That's why they are hydrophobic, because you have a long hydrocarbon chain. Only one carboxyl group is there, which is not sufficient to keep that long hydrocarbon chain soluble. So that's why they will bind with albumin for transport process. And MCFAs, SCFAs, they pass through cell membrane and mitochondrial membrane and they are activated in the matrix of mitochondria. And LCFAs, they undergo free diffusion over cell membrane and they are activated over outer mitochondrial membrane. Now, here is how fatty acids are activated. So the figure here, it is showing activation of fatty acid. Now the fatty acid has to be attached with the CoA to activate it. So here is the fatty acid and this is the activated fatty acid, that is fatty acyl-CoA. So how fatty acid is converted into fatty acyl-CoA? The figure here is showing you that it is a fatty acyl-CoA synthetase enzyme is the one that will activate a fatty acid into its CoA form by using a reduced coenzyme A, that is shown here. And note that reduced coenzyme A, it is a derivative of pantothenic acid. In order to activate a fatty acid into fatty acyl-CoA, there is a consumption of ATP which is converted into AMP. So conversion of ATP into AMP, it is equivalent to consuming 2 ATP molecules. That's why for activation of a fatty acid, we need to consider there are 2 ATPs being consumed. So this information is necessary for us to calculate net number of ATPs that will come from oxidation of fatty acids. Later, we are going to use that information. Make sure you remember that. Okay. Now the activation of free fatty acids from the previous figure. So this is just a recap of your previous figure. Acyl-CoA synthetase is the enzyme that will activate a fatty acid, and this enzyme, what it does, it simply adds CoA to a fatty acid. Now we have different types of acyl-CoA synthetases. Acyl is just a general name of the enzyme there. It is acyl. Acyl can be any fatty acyl. Remember, fatty acyl. When I write fatty acyl, I am not mentioning a number of carbon in that particular fatty acid. It is just a general fatty acid. It will have N carbon, N number of carbon. We don't know what is that N. So, if it is a short chain fatty acid or a medium chain fatty acid or long chain fatty acid or very long chain fatty acid, so each fatty acid has their own fatty acyl-CoA synthetase. Just to give you an example, I am interested in a palmitic acid, palmitate. So, which is a 16 carbon saturated fatty acid, palmitic acid. So, I want to activate palmitic acid. What is the enzyme that activates palmitic acid? So, the enzyme is palmityl-CoA synthetase. Palmityl-CoA synthetase, because the fatty acid that is activated is palmitic acid. That's why the name of the enzyme is palmityl-CoA synthetase. That's the enzyme. This is a specific enzyme like that. So, if my fatty acid is stearic acid, the enzyme name is stearyl-CoA synthetase, right? So, like this, depending on the type of fatty acid, that specific fatty acyl-CoA synthetase will be present. So, what I am trying to explain here is every fatty acid has its own acyl-CoA synthetase. So, for medium chain fatty acids, we have medium chain acyl-CoA synthetase. For short chain fatty acid, we have short chain acyl-CoA synthetase. Both of these category are there in the matrix of mitochondria. Whereas long chain acyl-CoA synthetases, they are present over outer mitochondrial membrane. Remember that. So, short and medium chain fatty acid acyl-CoA synthetases are present in the matrix of mitochondria, whereas long chain fatty acyl-CoA synthetases are present over outer mitochondrial membrane. They are present over endoplasmic reticular membrane, and also they are present over peroxisomal membrane. Now, take a note of the different locations of long chain fatty acyl-CoA synthetases. They are present in 3 locations. One is in the outer mitochondrial membrane. So, the long chain acyl-CoA synthetases being present over outer mitochondrial membrane, so they are going to allow transport of long chain fatty acids into the matrix of mitochondria for beta oxidation process, and thereby a cell can get energy. That's the purpose there. What is the purpose of long chain acyl-CoA synthetases being present over endoplasmic reticulum? It's because whenever there will be accumulation of long chain fatty acids, maybe because of saturated beta oxidation process, at that time some of the long chain fatty acids are diverted into endoplasmic reticulum for esterification. And also, when long chain fatty acids are synthesized in the cytoplasm in well-fed condition, these long chain fatty acids are diverted into endoplasmic reticulum for esterification with glycerol 3-phosphate to make triacyl glycerol synthesis. So, triacyl glycerol synthesis is going on in the smooth endoplasmic reticulum, and for that you need long chain acyl-CoA synthetase, because activated fatty acid is the one that undergoes esterification. Whereas in the peroxisomal membrane, presence of long chain fatty acyl-CoA synthetase is to activate a fatty acid that gets into plasminogen synthesis or oxidation of long chain fatty acid in the peroxisome. Like this, depending on the different functions of these subcellular organelles, so long chain fatty acyl-CoA synthetases are present there. So, over outer mitochondrial membrane, endoplasmic reticulum and peroxisomal membrane. Now, let's go on to see a very long chain fatty acyl-CoA synthetase, which is present over peroxisomal membrane, and that is for oxidation of very long chain fatty acids. So this is how a different type of acyl-CoA synthetases are present for each type of carbon length fatty acid. So we have short chain fatty acyl-CoA synthetases, medium chain fatty acyl-CoA synthetases, long chain fatty acyl-CoA synthetases, and very long chain fatty acyl-CoA synthetases. Now let's move on to see a carnitine shuttle mechanism. Now the carnitine shuttle mechanism is that it helps in the transport of long chain fatty acids through the inner mitochondrial membrane for beta oxidation process. Carnitine shuttle mechanism is not required for short and medium chain fatty acid, at least in the mitochondria. It is required in peroxisomal membrane because when the peroxisome is oxidizing very long chain fatty acids into short and medium chain fatty acids, so to transport short and medium chain fatty acids out of the peroxisomal matrix into the mitochondrial later, so there we need a carnitine shuttle mechanism because they are carried by binding to carnitine. But here what we are seeing is the mitochondria. This is the mitochondria. So this is mitochondrial carnitine shuttle mechanism for a long chain fatty acids, LCFAs. The fatty acid that is mentioned here in the figure is the long chain fatty acid. Now let's get into the details. What is this carnitine shuttle mechanism here? So as you can see here, over outer mitochondrial membrane, so there are two enzymes. One is acyl-CoA synthetase, which is a long chain acyl-CoA synthetase. So because we are looking at transport of long chain fatty acid across the mitochondrial membrane. So we have acyl-CoA synthetase on the outer mitochondrial membrane, long chain acyl-CoA synthetase, which is going to add CoA to a long chain fatty acid to make it as long chain acyl-CoA. Now this long chain acyl-CoA passes through outer mitochondrial membrane into the intermembrane space. Now in the intermembrane space where the carnitine is also available there, and this carnitine, before I take you into further details, I want to explain you how carnitine is synthesized and being made present there. This carnitine here, it is synthesized in the liver. Predominant organ that synthesizes carnitine is liver. And in the liver, carnitine. Now let's move on to see carnitine shuttle mechanism. Note that carnitine shuttle mechanism is necessary for transport of long chain fatty acids through the inner mitochondrial membrane to go into matrix of mitochondria for beta oxidation. Now before I explain you carnitine shuttle mechanism, so let me explain you what is carnitine. The carnitine, it is synthesized in the liver. Liver is the major organ where carnitine is synthesized, and the carnitine synthesis, it needs two essential amino acids. One is lysine and other is methionine. Now what happens in the liver is lysine, it is methylated to become trimethyl lysine, and that uses these two amino acids, and that is lysine and methionine. These two are the essential amino acids that are required for the formation of trimethyl lysine, and which is eventually converted into carnitine. And in this process, there is an enzyme called gamma-butyrobutane dioxygenase. This is a four-step process. Carnitine biosynthesis involves four enzymatic reactions. One of the enzymes that I would like to mention here is gamma-butyrobutane dioxygenase. Let me write it down. The name of the enzyme is gamma-butyrobutane dioxygenase. This gamma-butyrobutane dioxygenase is one of the enzymes involved here in the carnitine biosynthesis. Now, why we need to know this enzyme name? And also why we need to know the requirement of carnitine biosynthesis, that is lysine and methionine, essential amino acids. It's because if the person has got essential amino acid deficiency, especially lysine and methionine deficiency, that will lead to decrease in carnitine availability, and decrease in carnitine availability means carnitine shuttle mechanism may not function optimally. That's one thing. The second thing is there is a drug called meldonium. Let's get into that. So the drug meldonium, this meldonium is a doping drug which is used by sportsmen. So what it does? So the doping drug meldonium, it is a modulator of L-carnitine metabolism. It's an L-carnitine metabolic modulator. So what it does, it is going to inhibit gamma-butyrobutane dioxygenase. That is why I was mentioning about this enzyme here. Gamma-butyrobutane dioxygenase is inhibited by meldonium. And because it inhibits gamma-butyrobutane dioxygenase, which hydroxylates butyrobutane into carnitine. So this is the enzyme that is helping in the synthesis of carnitine, and that is inhibited by meldonium. And also meldonium, not only it inhibits the synthesis of carnitine, it also inhibits L-carnitine transmembrane transport of long-chain fatty acids via a transporter called SLC22A5. SLC22A5. This is the membrane transporter which helps in the transport of carnitine across the membrane in the peripheral cells, so that peripheral cells can use this carnitine for transport of long chain fatty acids across inner mitochondrial membrane. So that is also inhibited by meldonium. So the meldonium has two effects. One thing is it is going to decrease synthesis of carnitine by inhibiting gamma-butyrobutane dioxygenase enzyme, and the second thing is it is going to inhibit a carnitine transporter to the peripheral cells over plasma membrane called SLC22A5. So by doing this, overall meldonium, it is going to decrease availability of carnitine for carnitine shuttle mechanism. So thus the decreasing L-carnitine levels in the tissue and plasma decreases fatty acid oxidation and thereby mitochondrial ATP generation. And this way, meldonium, it causes cardiac reconditioning by diverting ATP production from oxidation of fatty acids to glycolysis. Because oxidation of fatty acids, fatty acid oxidation is the one that will give major energy during exercise to the skeletal muscle and cardiac tissue. So when meldonium is inhibited, so the long chain fatty acids which need to go into the matrix of mitochondria for beta oxidation process and to provide energy, and that is now decreased, and now the tissue like skeletal muscle and cardiac tissue, it will divert its fuel from fatty acids to glucose, that is the glycolysis. And in terms of oxygen requirement, whenever fatty acid oxidation is going on, fatty acid oxidation consumes much higher oxygen than the glycolysis. So that's why here by cardiac reconditioning, so consumption of oxygen is kind of reduced by diverting from fatty acid oxidation to glycolysis. That means when oxygen consumption is reduced, so that means the respiratory rate will also be reduced, and by reducing or decreasing or controlling the respiratory rate, so the exercise performance can be improved. And that's how sportsmen try to dope this drug meldonium to improve their sporting performance. And that's why this is one of the banned drug, meldonium. So this has shown, meldonium has shown beneficial effect in the ischemic conditioning of heart, cerebral ischemia, prevention of atherosclerosis, and also even prevention of diabetes mellitus by decreasing insulin resistance. So not only it is useful in increasing the exercise performance, but it has got a beneficial effect in treatment of certain diseases like ischemic heart conditions, cerebral ischemia, prevention of atherosclerosis, and also in prevention of diabetes mellitus. So that's something about meldonium that you should be aware of. So with that information, where I have explained you how carnitine is synthesized and where it is synthesized and what are the two essential amino acids that is required there and what are the clinical application of that carnitine concept, especially with the meldonium. Okay, now let's get back to carnitine shuttle mechanism. Now, as you see here in this figure, long chain fatty acid activated over outer mitochondrial membrane into fatty acyl-CoA, and that fatty acyl-CoA in the intermembrane space, it is condensed with carnitine. This is done by an outer mitochondrial located enzyme called carnitine palmitoyl transferase 1 enzyme. And now this carnitine palmitoyl transferase 1 enzyme is also called as carnitine acyl transferase 1 enzyme. So that is, other name is carnitine acyl transferase 1, that is C81, CAT1, carnitine acyl transferase 1. So carnitine palmitoyl transferase 1 is a specific enzyme specific for palmitic acid. If fatty acid, if you don't know the number of carbons in a long chain fatty acid, it can be written as carnitine acyl transferase 1. So that's another way of writing CPT1 there. Okay, so which is present over outer mitochondrial membrane, and this enzyme, what it does, it is reversibly esterifying carnitine to fatty acyl-CoA. It's a reversible reaction where carnitine is attached to the hydroxyl group of carnitine is attached to a fatty acid to make it as fatty acyl carnitine, and this fatty acyl carnitine is now getting down into the matrix of mitochondria by an anti-porter system called carnitine acyl carnitine translocase. And once it is inside the matrix of mitochondria, fatty acyl carnitine, it will be reacting with the another enzyme which is located in the inner mitochondrial membrane called carnitine palmitoyl transferase 2 enzyme. This is also can be written as carnitine acyl carnitine transferase 2 enzyme. Okay. Now, what this CPT2 enzyme does, it is going to remove carnitine from that fatty acyl carnitine and release that as free carnitine, and fatty acyl-CoA is made. So, CoA is added, carnitine is removed. So, we have fatty acyl-CoA in the matrix of mitochondria, which is now ready for beta oxidation in the matrix. Remember, beta oxidation is a mitochondrial matrix reaction. Now, what happens to the carnitine that is free here in the matrix of mitochondria? It will come back into the intermembrane space by that anti-porter system, that is carnitine acyl carnitine translocase enzyme, and that is going to bring one more fatty acyl-CoA in the form of fatty acyl carnitine into the matrix of mitochondria. So, every time carnitine become free, it will go back to the intermembrane space, bring one more long chain fatty acid. That's why carnitine is moving back and forth here between matrix and the intermembrane space. That is why the name of this system is called as carnitine shuttle mechanism. So, what is the use of carnitine shuttle mechanism? It is going to help in the transport of long chain fatty acids from the outer mitochondrial membrane all the way to the matrix of mitochondria. So, that's the purpose of carnitine shuttle mechanism. Okay, so make sure you remember, and also CPT1 and CPT2, all these are reversible enzymes. So, they can reverse the reaction and separate carnitine and release fatty acid also. So, remember that point also. Now, alright, now let's get into clinical aspects related with carnitine shuttle mechanism. So, the first disease here is carnitine deficiency. You know now the importance of carnitine. So, carnitine is necessary for the transport of long chain fatty acids into the matrix of mitochondria, and also you know carnitine is synthesized in the liver, and for synthesis of carnitine, now I already told you there is a requirement of, so first thing is liver, carnitine is synthesized in the liver. Just to recap, carnitine, it is synthesized in the liver, that's an organ. and then it needs lysine and methionine. Okay, so and also carnitine has to be transported from the liver to the peripheral cells in the blood, and in the peripheral cells it has to be taken up by the peripheral cells through the membrane, and the transporter is SLC22A5. This is the name of the carnitine transporter present over plasma membrane. So, and also carnitine, it's a small dipeptide. It can be filtered by the kidney just in case of renal conditions. There can be loss of carnitine in the urine. So, renal diseases, it can be affected. So, we'll have to look at the role of kidney there. Now, let's look into the disorders with that background information. So, let's look into carnitine deficiency. So, the carnitine deficiency can be divided into primary carnitine deficiency because of the genetic condition, a mutation in a gene that is coding for SLC22A5, a membrane transporter. It will lead to decreased availability of carnitine in the peripheral tissues, and that will lead to carnitine deficiency. And the second is acquired carnitine deficiency. So, we have seen the primary carnitine deficiency. Second is acquired carnitine deficiency. Acquired carnitine deficiency can be because of deficiency in the essential amino acids, that is lysine and methionine. You know why lysine methionine deficiency can lead to carnitine deficiency? Because lysine and methionine, these are the two essential amino acids required for carnitine biosynthesis in the liver. So, any conditions that affect liver can lead to carnitine deficiency, like liver diseases can lead to impaired synthesis of carnitine in the liver, or it can be loss of carnitine during hemodialysis process or renal failure conditions. So, hemodialysis and renal failure can also lead to carnitine deficiency, and also intake of drugs like valproate, pyrampicillin, emetic, zidovudine, all of them can lead to acquired carnitine deficiency because of loss of carnitine. Okay, so what is the effect of carnitine deficiency? Because carnitine is required for a transport of long chain fatty acids across the inner mitochondrial membrane. So, the deficiency of carnitine will lead to decreased oxidation of long chain fatty acids in the matrix of mitochondria. So, that's what I have written here. So, decreased LCFA oxidation, because LCFA oxidation is decreased, so that will lead to accumulation of long chain fatty acids in the tissues, leading to increased levels of long chain fatty acids in the tissues which are building up. And these long chain fatty acids, because they are accumulating in the tissue, so they can be diverted into endoplasmic reticulum, and in the endoplasmic reticulum they can be converted into something called as dicarboxylic acids. Always remember, whenever fatty acids are not undergoing oxidation and whenever fatty acids are building up, and these fatty acids which are long chain, so they will be undergoing a process called as omega oxidation, and this omega oxidation will be going on in the endoplasmic reticulum. And in the omega oxidation, what happens is, let me write down a short form of fatty acid is CH2 N number, COO minus on one side and CH3 on this side, methyl group. Every fatty acid has a carboxylic group and a methyl group. In between you have a hydrocarbon chain. So, in the endoplasmic reticulum, what happens? The methyl group here, this methyl group here, that's the omega carbon, will undergo oxidation into carboxylate group, COO minus, and then we have CH2 N times and COO minus. That means we have two carboxylic groups here. One carboxyl group on the left side, another carboxyl group on the right side, and this type of fatty acid means organic molecule, we call it as dicarboxylic acid. Dicarboxylic acid, two carboxylic acids on either side, right? So, that is what happens to long chain fatty acids whenever there is a decreased fatty acid oxidation. So, some of the fatty acids diverting into endoplasmic reticulum are converted into long chain dicarboxylic acid. And also, since carnitine is not available sufficiently, we are looking at carnitine deficiency. We don't find a normal number of acyl carnitine. You know, fatty acyl carnitine, CPT1 adding carnitine to fatty acid to make fatty acyl carnitine. So that kind of fatty acyl carnitines will be low, okay, in the blood and urine, and the patients will show certain signs and symptoms because of this. And also in carnitine deficiency, blood levels of carnitine will also be decreased in carnitine in the blood. Decreased carnitine in the blood can also be a laboratory feature. So let me just finish laboratory feature and then going into clinical or other conditions that can be seen in carnitine deficiency. So one is decreased LCFA oxidation leading to increased accumulation of LCFA, leading to formation of more long chain dicarboxylic acids and low levels of acyl carnitines and low levels of carnitines in the blood. Now because of this lack of oxidation of long chain fatty acids, so ATP needs of gluconeogenesis will be decreased. So because you know for gluconeogenesis fatty acid oxidation has to go on, because the ATP needs of gluconeogenesis has to come from oxidation of fatty acid. When the oxidation of fatty acid is down here because of carnitine deficiency, it will lead to decrease in gluconeogenesis process under fasting condition, and that will contribute to hypoglycemia that is seen in carnitine deficiency. For that matter, any carnitine deficiency or carnitine shuttle mechanism defect or even beta oxidation defect will lead to hypoglycemia. And also patients with carnitine deficiency may show hypoketonemia, means lack of ketone bodies under fasting condition, because normally under fasting condition beta oxidation will go on, acetyl-CoAs are made and some of the acetyl-CoAs are diverted into ketone bodies in the matrix of mitochondria. Ketone bodies will appear in the blood whenever the person goes into fasting condition for a prolonged period of time, and that finding you don't see here. In fact, because of lack of oxidation of fatty acid, lack of acetyl-CoA, which are not coming from oxidation of fatty acid, so you don't see a ketone bodies appearing in the blood even after fasting condition. That is what is called as hypoketotic and hypoglycemia. So low or no ketone bodies seen in the blood along with the low glucose level. So that is what is one of the important finding of carnitine deficiency or any carnitine shuttle mechanism defect including beta oxidation. Remember that point. Hypoketotic hypoglycemia, one of the common sign that you are looking for in carnitine deficiency, CPT1 deficiency, CPT2 deficiency and defect in beta oxidation. All of them. Make sure you remember, these patients will show hypoketotic hypoglycemia. Why? Because when the beta oxidation is not going on or long chain fatty acids, wherever the defect can be, so low acetyl-CoA levels means low ketone bodies. And also when beta oxidation is not going on, ATP levels are not sufficient to maintain gluconeogenesis. So patient will go into hypoglycemia. So that's a common finding. Along with that, patient will show encephalopathy, brain is affected, cardiomyopathy, cardiac tissue is affected, skeletal muscle myopathy, skeletal muscles affected, hepatomegaly, liver being enlarged, elevated liver transaminases indicating there is abnormalities in the liver going on, hyperammonemia because liver is failing, urea cycle is affected leading to hyperammonemia, recurrent episodes of abdominal pain, diarrhea, hypochromic anemia and recurrent infection. All these are the signs of carnitine deficiency directly or indirectly related with a lack of energy coming from oxidation of fatty acids. Treatment is, you need to address the underlying cause and whether it is especially in the acquired carnitine deficiency. If it is an essential amino acid deficiency, it will have to supplement essential amino acid lysine and methionine. If it is a liver disease, you will have to address liver condition. If it is a renal condition, address the renal issue. Or if it is because of drug like valproate, pyrampicillin, emetine, zidovudine, this kind of thing, you will have to address that particular cause. And then we can give oral carnitine supplementation to improve the condition. So that's about carnitine deficiency. Now let's move on to see carnitine palmitoyl transferase 1 enzyme deficiency. Let me first show you the figure and then I will explain you I will give you the details in the next slide. So what I am doing now is I am going to block this enzyme. Okay, carnitine palmitoyl transferase 1 enzyme is not working, deficient, mutated, gene is mutated, enzyme is defective. So what will happen? So obviously fatty acyl-CoAs will be elevated, right, and the carnitine levels will be elevated because it is not being used. So fatty acyl-CoAs will come into cytoplasm. So we have especially long chain fatty acids, because these are all LCFAs, long chain fatty acids coming into the cytoplasm, and these long chain fatty acids will go into the blood. So they will be you are going to find them at increased levels in the blood, and also long chain fatty acids can go into endoplasmic reticulum ER, undergoing omega oxidation and becoming long chain dicarboxylic acid. Long chain dicarboxylic acids you are going to find in the blood. Say they will appear in the blood and also they will appear in the urine. And also note that this free carnitine which is elevated, that will also appear in the blood. So there will be increase in blood levels of free carnitine because they are not consumed. Carnitine levels will be elevated in the blood and also in the urine. So, and what about fatty acyl carnitine? So what about this fatty acyl carnitine here? So CPT1 is down, means fatty acyl carnitine synthesis will be decreased. That's why blood levels of fatty acyl carnitine you are going to see decrease. Fatty acyl carnitine will be decreased. So decrease in fatty acyl carnitine levels. So these are the findings that you see in CPT1 deficiency. So that is increase in LCFAs in the blood, increase in long chain dicarboxylic acids, increase in free carnitine levels in the blood and urine, and decrease in acyl carnitine levels in the blood. Long chain fatty acyl carnitine levels will be decreased because the enzyme that is esterifying carnitine with the fatty acyl-CoA itself is decreased. That's why you see the decrease in fatty acyl carnitine. That's most important information that you need to remember to diagnose CPT1 deficiency, at least in the case 10. Okay, remember that. So here are the findings. So in CPT1 deficiency, decreased LCFA oxidation leading to accumulation of LCFA in tissues, leading to increased levels of LCFAs, long chain dicarboxylic acid, increase in free carnitine, but most importantly, it is low levels of acyl carnitines in the blood and urine. What are all the clinical signs and symptoms? See, the signs and symptoms related with hypoglycemia can be seen because patients will show hypoketotic hypoglycemia. I already told you that any carnitine defect, including beta oxidation, will lead to hypoketotic hypoglycemia, and sometimes it will be mentioned as non-ketotic hypoglycemia. Hypoketotic hypoglycemia or non-ketotic hypoglycemia. So hypoketotic hypoglycemia or non-ketotic hypoglycemia is one of the important signs that you see in carnitine shuttle mechanism defect or in beta oxidation defect. Along with that, patients will show cardiomyopathy, skeletal myopathy, encephalopathy, impaired liver function and all that. Okay, so look for the laboratory features, and a key sign of beta oxidation defect or carnitine shuttle mechanism defect, that is hypoketotic hypoglycemia. Cardiomyopathy, skeletal myopathy, encephalopathy, they can be commonly seen in most of these carnitine shuttle defects or beta oxidation defects. So that can be common finding there. So look for acyl carnitine levels also to differentiate between CPT1 and CPT2 deficiency and beta oxidation being defect. So in CPT1 deficiency, importantly, so you don't low levels of acyl carnitines you are going to find there. Now let's move on to see CPT2 enzyme deficiency. Let's block this CPT2 now. So CPT2 is not working. So when CPT2 is not working, so there will be elevation of fatty acetylcarnitine in the matrix of mitochondria. And these fatty acetylcarnitines, what they will do, they will damage the inner mitochondrial membrane. So inner mitochondrial membrane will be damaged. So they will leak out into the intermembrane space. So their levels will be increased in the intermembrane space, and then they will damage outer mitochondrial membrane, and eventually you are going to find fatty acetylcarnitine in the cytoplasm, and then it will appear in the blood and urine. So blood and urine levels of fatty acetylcarnitine levels will be elevated. That's one of the important finding here with CPT2. And then what about carnitine levels? See these carnitines which are already attached with the fatty acetylcarnitine. So the carnitine levels will be down. So blood carnitine levels will be decreased. Free carnitine levels will be decreased. Some of the long chain fatty acids can come out into the cytoplasm. So LCA phase comes out because of damage to the mitochondrial membrane. So you will see increase in LCA phase and also increase in LC long chain dicarboxylic acids, because some of the long chain fatty acids will go into endoplasmic reticulum to become dicarboxylic acid. So these are the findings in CPT2 enzyme deficiency. So that is most importantly increase in fatty acetylcarnitines, decrease in free carnitines, increase in LCFA, and increase in long chain dicarboxylic acid. So that is what I have written here. So look at the details. So decreased LCFA oxidation is seen in CPT2 deficiency, accumulation of LCFA in tissues, increased LCFAs, increased long chain acetylcarnitine. See, that's the important point. Increase in long chain acetylcarnitines is the differentiating point there, and then increase in long chain dicarboxylic acids. And the feature that I already explained you is hypoketotic hypoglycemia, along with signs of cardiomyopathy, skeletal myopathy, encephalopathy, impaired liver function can be seen. Out of CPT2 deficiency in different tissues, the most common CPT2 deficiency is seen with muscle-specific CPT2 enzyme deficiency is more common. So since muscles are affected here, so the patients with CPT2 deficiency here will show exercise intolerance, muscle tone, muscle ache, mild to moderate muscle weakness, rhabdomyolysis, myoglobinuria, and when the muscle will break open, it will give rise to increase in the levels of inosine, hypoxanthine, uric acid, hyperammonemia. All these signs can be seen, right? And the treatment is got to give IV glucose just to decrease the chances of fatty acid oxidation in the cell and cease muscle activity, just so that rhabdomyolysis is not going on, and thereby decreasing the signs and symptoms. So this is what is carnitine palmitoyl transferase 2 enzyme deficiency. So that's about mobilization of fatty acids from the adipose tissue, and then the transport of fatty acids, especially long chain fatty acids, from the adipose tissue to peripheral tissues, and then transport of activation of long chain fatty acids over outer mitochondrial membrane, transport of them into the matrix of mitochondria by carnitine shuttle mechanism, and the synthesis of carnitine, meldonium, role of CPT1, CPT2, deficiency of carnitine, CPT1 deficiency, CPT2 deficiency, all these things we have discussed. I hope this video has helped you in understanding this concept.